Hello everybody. I see some of my students here as well. I'm just going to give everyone some time to connect, just a couple minutes, so that they don't miss out if they were planning on joining. I am in South Africa at the moment. It's only 2 here. It's early. And I've come into school today. I'm doing this live from my classroom. It's quiet here. I won't get disrupted by anybody. But in the meantime, while we also wait for everybody to join, I'm just going to tell you a little bit about what we're going to do, and so you're a little bit prepared for it. So just a disclaimer as well for this video. This is just how I teach. Maybe your teacher has taught you something different. And that's fine in terms of how they've approached their work. This is how I teach my students, so my students will be very familiar with what we're going to be doing now and how I make my notes. I'm going to upload this video once we're done to YouTube, and then I'm going to timestamp the different topics so you can just fast forward through to those parts, especially if you want to make a note on things that we've done. Yeah, and then the things that I want to cover today, there's some specific things that I want to cover. So I want to cover things that are always really, really badly answered. Things like the female reproductive hormone cycle, the menstrual cycle, meiosis, easy topic, really poorly answered, like really poorly answered by Matrics. And then I'm just going to open it up to you, and you can ask me some questions if you want. Yeah, and I think I'm not going to wait too much longer. Let's get into it. I'm going to swap my camera around so that you can see the page that I'm working with. Yeah, Ntando, I see you sharing my live. Thank you. What was I about to say now? Oh, so let me get into some important things that you are going to need. I hope that everybody here who's in Matric has this document from the government. I know it's probably backwards to you, but this is what's called the exam guideline. I know it says 2017, but this is when it was written. It still applies to 2020. If you don't have this document, you're playing around. You have to have this in order to write the final exam. Basically what it is a detailed description of all the topics that you need to know, what you can leave out. Some textbooks put way too much information into their textbook, and you think, do I have to know all of this? The answer is no, you don't need to know all of it. So this is a really useful tool. If your teachers never introduced this to you, then you need to go and get it. And you can get this off of the government website, the education website. It's very easy to find. That's a very important tool. And then the second important tool is you need to get past papers. I would get consecutive papers. So I would get papers that are like 2018, 2019. I wouldn't do like 2016 and 2019. And the reason for that is you want to do consecutive papers because consecutive papers allow you to see what the trend was in the way they're asking the question. Okay, so I'm going to let you have a look at a note that I'm going to make now. And like I said, if you want to ask questions, I'll be able to see what you're asking, but I'm going to flip my camera around. So let's get into it. I'm going to do a quick note on the female hormones and the female reproductive system. Okay. So I think the issue with this particular topic is we don't know what hormones are doing what, and we don't know who is doing what. So this is how it goes. We have two sets of hormones, right? We have the pituitary hormones, and we have the ovarian hormones. These are the two sets of hormones, right? Then under the pituitary hormones, we have FSH and LH. Everyone should know what these mean. Follicle stimulating hormone, luteinizing hormone. Then for the ovarian hormones, we have estrogen and we have progesterone. Okay. All right. So often we get confused with what they all do, and so I'm going to quickly describe to you what they all do and how they all work. So first of all, let's number these out and how they work. So in order for the female system to work, we need to start off with follicle stimulating hormone. This is where we begin. Okay? And follicle stimulating hormone stimulates a follicle to be released. So what I'm going to do is I'm just going to box these off so I can keep them separate quickly. Right. So the follicle stimulating hormone stimulates a follicle to be produced. So here's my follicle, and that is then sent to the ovary, and the ovary starts to mature a follicle. Now, as this follicle grows, it starts to secrete estrogen. Now, estrogen is an ovarian hormone, and it has a specific function. So what I'm going to do down here is I'm going to list the functions quickly of each of these two hormones. So FSH and its function is to stimulate the production of a follicle, right? That's what its name is. Then on estrogen side, estrogen's job is to regrow, and this is important, regrow and thicken the endometrium. Okay. Often estrogen and progesterone are confused with one another in terms of what their actual jobs are, and we're going to make sure that by the end of this lesson you'll know very clearly what the difference is between the two. So if we go back up to our little sketch, FSH has stimulated our follicle, and this follicle, as it gets bigger, it secretes estrogen. And estrogen's job is to regrow and thicken the endometrium. Remember, we lost the endometrium at the end of our last cycle. So how do they ask this kind of question, and why is it so difficult? Well, generally they give us some kind of like graph in a question. And, someone's asking, do we say lining or endometrium? Tando, you need to say endometrium, please. Don't say lining. It's important to use your vocabulary, everybody. You might be marked down if you don't use the correct vocab. So effectively, on my little graph here, I have day 0, 14, and 28. These are milestones, right, in time. And so if we start off on day 0, I'm just going to use different colors to represent different hormones. Let's go with FSH. So how do we identify FSH on our graph? So FSH, and I want you to know that all these hormones never start off at 0. It gradually increases in the beginning, and then there will be a dramatic peak. Okay, this peak signifies the acceleration of creating a new follicle. However, it will drop off, right? This is FSH on our graph. Now, if I were to include estrogen, I'm just going to use a different colored pen. Estrogen, on the other hand, is also going along, not much increase, and it will increase after FSH has peaked. So FSH peaks, and shortly after FSH peaks, estrogen has a peak. Right? And that's how I'm going to leave this graph for now, because I'm going to expand it as we go along. Okay? So for right now, for this moment, and for this instance, we're only working with these two hormones. So let's go back up to our diagram. Estrogen was secreted. It's thickening and regrowing the endometrium. What are we preparing for? We are preparing for ovulation, and we are preparing for pregnancy. So as the follicle develops and gets bigger. We need to now release it. And this is where luteinizing hormone comes in. This is the third hormone we need to introduce. This hormone is going to prepare our follicle. But I'm going to pause here for 2 seconds. Do you agree that if we've grown a follicle, do we need to grow another follicle? In other words, can I be pregnant at 2 different stages? In other words, you're 6 months pregnant and then your other fetus is 1 month pregnant. The answer is you can't be at 2 different stages. So the only way to stop this is for estrogen to have a negative feedback on FSH. So this is negative feedback. Okay? All right. Now, estrogen puts a negative feedback on FSH, which means our FSH levels drop, which is what I drew over here. So that's why it drops. And yes, they can ask that. Now, once FSH is slowly decreasing, LH is going to start to be secreted. And LH is sent to our follicle. And let me just label it follicle, so we don't forget what that is. And luteinizing hormone, its effective job is to mature the follicle and eventually release it. Now, once luteinizing hormone has been added to this, we are going to end up having the follicle burst, which means we ovulate. And the follicle turns into the corpus luteum. I'm just going to write that up here. Corpus luteum. Right? Our corpus luteum. I think I've actually spelled that wrong. Whoops. Luteum. There we go. Okay, so this is the corpus luteum. Now, the importance of the corpus luteum is to secrete, and I'm going to try and figure out how I'm going to do this without covering all my labels, to secrete progesterone, which is our 4th and final hormone. Progesterone is the pregnancy hormone, right? So, effectively, progesterone's purpose is to prepare for pregnancy, right? But it does this in a very specific way. Progesterone affects the endometrium. Remember, these are all ovarian hormones, which means they affect the ovarian cycle and they affect the endometrium lining. And so progesterone maintains, and this is so important, this is not the same as regrow and thicken, maintains and makes more vascular. Vascular, if we're not sure what that means, when you think of vascular, you must think of blood vessels. Effectively, we're making it more filled with blood vessels in order to support a pregnancy, right? Okay. So, now we have all of our hormones in play. Let's quickly go down to our graph and let's add them in here. So, luteinizing hormone is quite a low hormone throughout this whole process, and it's going to go through fairly low until we get to day 14, which remember is ovulation day, right? That's when we ovulate. And so it's going to peak on ovulation day, big peak, and then it's going to drop off. And there's no need to keep it very high because once you've ovulated, you've ovulated, right? You don't need to keep this hormone very high up, and it will drop off. An easy way to identify FSH is it's the only hormone that peaks and then drops off on day 14. That's it. It doesn't come back again in a high amount at any point. But what's happening with our other hormones? So FSH actually is also responsible for a little bit of ovulation. So it does a little peak and then it too drops off. And there's no need for it to increase over the coming weeks simply because we're not growing another follicle. We want to see if this follicle we made is actually going to take hold and it's going to grow, right? So what's happening with estrogen? So estrogen at this point, it peaks just before ovulation. That makes sense. Why? Because we're preparing the endometrium for implantation. We're preparing it to regrow and thicken, right? So it makes sense that it peaks just before this. Then what happens is it also takes a little bit of a drop as well because it's done its job. And why does it take a drop? Because the follicle no longer exists. Because why? The follicle burst. Remember, it burst. It's not there anymore. It's now the corpus luteum. Now, let's add in progesterone. Progesterone is the pregnancy hormone, and if we're looking at our graph here, it too is a fairly stable hormone in terms of what it looks like on this graph. And it too sort of stays very stable, quite low at the beginning. And then after day 14, remember after ovulation, it will start to dramatically increase in amount. Why? Because the corpus luteum now exists. The corpus luteum now exists, and what that means is it will secrete progesterone. So let me just quickly recap. On the day of ovulation, FSH and LH are going to peak. Why? Because we're releasing the ovum. Estrogen takes a bit of a dip because the follicle no longer exists, so it's not going to secrete as much estrogen, so that drops off. In terms of progesterone, progesterone is a very flat hormone, okay? It doesn't increase a lot, okay? And it slowly increases, and then there's a dramatic increase after day 14. The reason why it increases after day 14 is because we are preparing for pregnancy, and remember, what are we trying to do to the endometrium? We're trying to maintain it. We're trying to make it more vascular. We want to grow as many blood vessels as possible because that's obviously important for the development of the placenta. So now progesterone has increased. Okay, now go back to our hormones one last time. Estrogen and progesterone have a negative feedback on their pituitary hormones, and it's a very simple reason. There's a negative feedback because thank you everybody. Luanga, I see your message. That's LH. I beg your pardon. Sorry everybody, that's LH. Okay. Where was it now? Oh, maintains our vascular. So they have a negative feedback, right, on our pituitary hormones. Why is that? That is because these hormones are trying to make an ovum, an egg. These hormones are trying to get that ovum to grow into a baby. Now think of the logic. I can't be making a new baby if I'm already pregnant with one. I have to work on one at a time. Disregard the idea of multiple babies, disregard twins and all of that, because effectively, if you're having twins, they're growing at the same time, at the same rate. And so think of these two categories of hormones as separate effects. And they are what we call antagonistic hormones, everybody. Antagonistic hormones. That means that if one goes up, the other goes down. That's what negative feedback is. Okay? Wonderful. Right. So let's wrap up our graph so we can go on to a different topic now. This progesterone level, if we fall pregnant, and I'm going to do this as a dotted line, if we fall pregnant, it will remain flattened well past day 28, because obviously that's pregnancy, right? So I'm just going to put here in brackets, pregnancy. Okay? Let's say someone doesn't fall pregnant, right? Progesterone is going to start to decrease and then dramatically drop off. This peak to this drop off is what signifies menstruation. And I just want you to notice something. This is roughly day 21. It might be a little bit more than 21. This might be day 22, 23. But this signifies the beginning of what we would call menstruation. In other words, we're building up. It is pre-menstruation, so we're building up to the loss of the lining, to a period, basically. Now, I can't forget the rest of our hormones, right? So, what's happening with estrogen? Well, estrogen does this little weird thing where it does this little boop, a little extra increase, and then it too also drops off and starts to level down. And a nice way to remember this is these two hormones, when they peak together like this around day 21, they do a last little push to get rid of the lining of the endometrium effectively. And that's what signifies the beginning of menstruation. And so LH is a very low hormone, so I'm just going to draw that off to the edge. And let's finish off with FSH. Now, if we're coming to the end of our cycle, we are definitely not going to keep FSH this low. It has to return up again. You must remember, as the lining sheds, so will a new follicle grow. Remember, this is all simultaneous. In other words, we're going to go slowly creeping back up again. And then obviously this line picks up again on this side on that line. Okay. All right. I'm just going to flip you all around so you can see me for 2 seconds. So it doesn't look like we're always staring at a blank page. Are there any questions on female reproduction? Because I'm going to do meiosis next. So any quick questions that I can answer for you? No questions? It is a very difficult topic, everybody. It's actually one of the worst sections answered always. And I mark at the end of the year, which means I'm one of the people that may mark your paper, especially if you write in the Western Cape. I can try and do DNA replication. Auxins, that's a tough one. The chorionic villi. The function of chorionic villi, everybody, is to attach onto the placenta so that there's a larger surface area for gaseous exchange, for effectively waste exchange, and it allows for the placenta to attach. I am going to do the differences between meiosis 1 and 2. Let's see what else your questions were. Is it Rome? Birth control, is it similar to negative feedback? Yes. I'm just going to say yes. Let's see, a couple other questions. What are the antagonistic hormones? So FSH and LH effectively are antagonistic to estrogen and progesterone. There are a couple of others that we don't really need to know in grade 12. So I think I'm just going to leave it with those 4 and make it easy for you. Okay, so yes, Ethan, I'm at school on a Tuesday, on a Saturday. This live is going to be saved for later, everybody. I'm going to upload it onto my YouTube channel, and I'm going to put timestamps in the different topics so you don't have to watch all the in-between stuff. Okay, so I'm going to quickly do... who's asking that? We are basically doing paper 1 stuff. We can do paper 2 stuff as well. Can you please do, can you please do possible questions they might ask on this topic? Sure. Okay, let me quickly say this. They're going to ask you something on female reproduction, 100%, okay? And they're going to ask you a graph question, no doubt. How they're going to ask you this is a different question in terms of, are they going to ask you like to label something? They might. I mean, if I look at some of these old question papers like this one, they've asked them to just label the female reproductive system and to put in some functions, but that's really easy. That's not difficult. I think what you're asking me is, what's the hardest question they could ask you on this? The hardest question they can ask you is to describe how the hormones influence the uterine and ovarian cycles. And those are 2 different cycles, which we've actually just done now. Those are my 2 blocks. Okay. All right, everybody. I think what I'm going to do now is I'm going to cover meiosis quickly, because I know my matrics are a little bit bad at this. Sorry to out everybody there, but yeah, you guys aren't amazing at this. So I'm going to quickly flip you around so you can see my note. Okay, so there's meiosis. All right, so this is the problem everybody has. Nobody can tell the difference between meiosis 1 and meiosis 2, and it's actually quite straightforward. Many of us just simply know the phases. That's it, right? Well, ladies and gentlemen, it's actually quite simple to tell the difference between meiosis 1 and meiosis 2, okay? Now, effectively, what you're always going to be looking for is you are going to be looking for the structure of your chromosomes, okay? So for meiosis 1, you're always going to look for chromosomes that are doubled, and not only that they are doubled, that they are with their homologous partner, okay? So we have 2. That's how we know we're in meiosis 1, because we have 2 chromosomes, homologous pairs here. How do we know we're in meiosis 2 instead of meiosis 1? That's another easy thing to look for. You will only ever see one of the homologous partners, and that homologous partner or that individual chromosome will be becoming a single strand. So, are they in a pair? Meiosis 1. Are they in an individual, or are they becoming a single strand? Meiosis 2. Okay, let's actually see if you know what you're doing. Those of you watching, show me that you actually know the difference. Okay, these 2 cells are going through meiosis. I'm not going to tell you 1 or 2. Okay, these are just rough sketches. Right. Which phase is which? If this is diagram A, this is diagram B, which is which? How do I know what phase they are in? Are they anaphase, metaphase, telophase, etc.? And are they in meiosis 1 or 2? Remember, we're going to look for pairs. And we're going to look for individuals. A, meiosis 2, very good. There's our individuals. Remember, these are individuals, everybody. Do you see a partner? Is a partner touching? This is not its partner. This is a different chromosome. Yes, diagram A is metaphase 2. How do I know it's metaphase 2? Because they've lined on the equator. Because here are my poles with my centrioles. And then on this side, what's B? Layla, it's not meiosis 1, prophase 1. What is it? It is. It's not metaphase 1. What is it? They're lining up on the equator. They are in pairs. They are not being pulled apart yet. It's not anaphase. What's our rule? When they come in pairs, what is this? What is this? It's not anaphase 1. I'm not seeing the right answer, everybody. Mo, thank you. Finally, metaphase 1. Ladies and gentlemen, if we could not do that, we are not ready for meiosis in the final exam. I just want you to be prepared for that. This is the same phase. Both of these are metaphase, everybody. Except this is metaphase 2, this is metaphase 1. How do I know that this is metaphase 1? Because, look here, our chromosomes are in a couple. There are 2. They are touching. Remember the rule, if they have 2, we're in meiosis 1. How do I know this is metaphase 2? Because I have individual chromosomes here. They're not touching anybody. They are lining up at the equator. Okay. Cool. Right. So, that's how we tell the difference between phases. Now I'm going to give you a tricky one. Prove to me that you actually know what you're doing. Okay, what phase is this? I'll give you some time to think. Yes, this is bio, Jack. It's not telophase. Amani, yes, this is prophase 2. Thank the Lord, someone got it right. Let me tell you why it's prophase 2. Number 1, the first and most important thing is, remember, chromosomes pair in homologous pairs. Homologous pairs are either the same size or they carry the same genes. Are these 2 chromosomes the same size? No. Therefore, they cannot be homologous pairs. What are you going to do if they don't color anything in the final exam? They leave it blank like this. We don't know if crossing over has happened. Okay? Maybe they'll do this. And they'll color in a little bit like that to show you that crossing over has happened. But this is prophase 2. Okay. Another quick one. What happens, I'm just going to flip my page over. What happens if you get a question like this? They give you 4 cells. And my matrics know this little problem. Right? This is the end of meiosis, right? 4 cells. And they do something like this. Okay, each one of my lines represents a chromosome. I have a cell with 4, 3, 1, and none. And this is what they say to you. And yes, this is a past question, by the way. I'm not just making this up. They ask you, how many chromosomes were in the original cell? And they also tell you that non-disjunction has taken place. In other words, you've ended up with a cell that has nothing inside of it, and you've ended up with cells that don't have the correct amount. What are you going to do? You have to tell me the original amount. Difficult, right? It's actually not too difficult, and I'll show you the simplest way to figure this out. At the end of meiosis 2, you have 4 cells, always, right? All you have to do is count how many chromosomes are present. 1, 2, 3, 4, 5, 6, 7, 8. There are 8 chromosomes at the moment here, but we're not going to stop there. That's just a little piece of information on the side. Okay? Each one of these cells is supposed to have the same amount of chromosomes, and they don't. So, technically, if we were to redo this, there are my 4 cells, and I have 8 chromosomes to work with. What's 8 divided by 4? 2. That means every cell is supposed to get 2 chromosomes. Right? But now this is at the end of meiosis 2. They want to know the original amount, as in what did we start with? Okay? So, if I go backwards and I rejoin my chromosomes in their cells, and I go back, these rejoin with their partners like so. Okay? How many chromosomes are in here? Still 2. Still 2. Put these back together. How many are there now? 4. 1, 2, 3, 4. Remember, this is a whole chromosome. So what was the original amount? 4. They can ask very, 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 very difficult questions. Okay? Meiosis is often a series where people underestimate how difficult it can be. Okay. Any other questions for me on meiosis and how to tell the difference? Oh, I'll give you some useful advice. Make sure when you read the question that the question says in a human or in a female or a male. It must actually say that, because if you start talking about female and the question says this meiosis was taking place in a male, and you start talking about ovums and things, you're going to lose marks. If you start, and this is a big one, if they ask a question about non-disjunction, so when chromosomes don't separate correctly, and they just say it went through non-disjunction, and they say discuss the result of non-disjunction, please don't start discussing Down syndrome. They're not asking for Down syndrome. They just want to know what's the result. How did we get there? Only talk about Down syndrome if they ask for Down syndrome. Please, grade 12s, please. This is such a common mistake that I see every year, and it doesn't seem to go away. It's the interpretation of the question. Okay, I'm just looking at the time. We can go for a little bit longer. I don't want to make this video too long because there's quite a lot of stuff to remember and to go through. I think I might do just one more thing. I'm just reading a couple of your comments. So if you are rewriting out there, and by the way, there are 1 million people writing Matric this year. Normally it's about 500,000 people writing, so it's double the amount this year because of all the rewrites. I'm going to be making a whole series of YouTube videos that do a couple of different things. I'm going to do tricky questions, and I'm going to explain tricky questions. Oh, I've lost connection. Sorry, I just lost connection for a few seconds there. I'm going to do a whole series on tricky questions from past papers. I'm going to do advice for people who are rewriting. So there's going to be videos on that. So you can go into my YouTube. The link to my YouTube is in my bio on my TikTok. And I'm going to be uploading a lot of videos in there too. Right now, the videos that I've uploaded are just my online teaching videos. So those are the videos that I would have uploaded for my Matrics when we were in lockdown, and you're more than welcome to go and watch them. I think they're very helpful and they're concise. You don't need the textbook that I'm talking about. It's just reference to our Matrics and where they were looking for pages and things like that. But yes, one of the most important videos I'm going to do is helpful tricks and hints and questions that you might have overlooked in your past papers, especially if you're rewriting. That's a video that I'll be uploading very soon. Okay, so let us do maybe one more thing. I'm just going to have a quick look through your comments quickly. I know that you want me to do paper 2 stuff, everybody, because I can see you asking for human evolution. I can see you asking for genetics, human evolution. Ooh, I see a lot of genetics, graphs, genetics. Okay, I'll do some genetics, everybody, because it clearly is like a hot topic. Protein synthesis. How do you write a paragraph question? Caddy, is it Caddy or Katie? Paragraph questions. Depends if it's an explain or a describe question. I've made a TikTok on that. You can go and find it there. I explain the difference and how to write a paragraph on that. Yes, okay. Ntando, I see what you're saying about make a paper 2 second live. I will. I'm going to do a whole bunch of little bits and pieces on TikTok, and I'm going to upload them for everybody to go and watch and follow. I know that paper 1 bio is, I think, in a week or so's time, where I have it in my diary. Let me quickly look at it, or my calendar. It is on the 27th of November. So in actual fact, you have 2.5 more weeks to go. So you've got some time, everybody. I am going to do a paper 2 as well for you, and I will upload that as well. I'm going to look through here, the ear and the eye. I'm going to make lots of videos, everybody. Yes, Ntando, I will relax. I know you want me to do evolution, everybody, but I'm trying to keep this video on paper 1 topics only. But I promise, I see Luke, you're asking for the paper 2 second live. Of course, I will do that. I see some of you are grade 10s and 11s. Right now I'm just going to focus on the matrics, just because I think they need the help. User 42900, etc. Should we do full papers or per question? Full papers. And please study the memos. Don't just look at the memo and be like, oh, I got that right, I got that wrong. Please, you need to do the whole thing. Yes, I do. I upload all my videos onto YouTube. I'm going to upload this live as well. I am going to be uploading a whole bunch of grade 11 TikToks as well. I've filmed them already. They're in my drafts, so I haven't posted them. Okay, so Keeks, my channel is in my YouTube. It's in my bio and my TikTok. You can go find it there. There's the link. Do you really want me to do genetic crosses? Is that what you want me to do? I mean, it is a paper 2 topic. Okay, I got genetic crosses the most. So let me tell you something, everybody. I'm a pro at these. These are like what I majored in. So this is where it's at. This is my strongest topic. Most of paper 2 stuff is my strongest stuff. So I know you want me to do a dihybrid. It takes too long for me to draw a dihybrid. However, I've come prepared. I do have an already drawn one somewhere here that I can grab and we can go through. So what I'll do now is I'll flip the camera around and we'll do a quick cross. Okay, everybody? So I'm going to do a quick monohybrid. Monohybrids, everybody remember mono meaning 1, right? Okay, so let's make this nice and easy, and I'm going to do a complete dominant cross. In other words, one trait is dominant over the other, right? Okay, so there's a format to your answer. You have to answer it this way, otherwise you are not going to get the full marks, okay? So we always start in the margin, P1, which means parental generation 1. Whatever the cross might be, maybe it's about flowers, it's about textures, it's about something. Let's say, for example, it's fur color of cats. So when we write this out, we're going to go a black fur cat, and let's go white fur cat. Now, before I go any further, let me give you some more information on the complete dominance. So if this is my cat question, and I want to look at fur, right? We need more information on this. So let's assume that black is dominant, so it gets a capital B, and white is recessive, so that's a little b. Okay, they will give this information to you in the descriptor, the question, right? They'll tell you which one is dominant and which one is recessive. Okay, so if this is a black fur cat, let's assume in the question it said that it was a homozygous dominant cat. In other words, it's got 2 capital B's. They could have also said this is a purebred. Whenever you see purebred, you must think they are the same letters. We multiply that by a white furred cat, and white is recessive, so it gets 2 small b's. Okay? Right. Then you're going to write the word meiosis just under that. Okay? And then you need to draw your gametes. So in the side margin, we're going to write gametes. And then you're going to put your letters in your gametes. So you're going to divide these. And remember, you can only have one allele. This is an allele. The letter is the allele, everyone, if we can't remember. I put circles around mine, and this is a good habit to do because you don't want to draw your letters 2 on top of each other. Okay. So then we write the word fertilization. Okay. And then in the column, we're going to go F1. F1 means filial, offspring generation. Now I use a Punnett square. Maybe at your school you have been taught to use a line diagram, but I use a Punnett square, and this is the one that's most commonly used by everyone. So now I copy in my alleles. Okay, yes, Ntando, you can draw circles, no problem. Okay, so I'm going to take my B, my 2 big B's, and this is coming from my 1 cat, and I'm going to put it into my Punnett square. You must keep the letters together if they come from one individual. And then we do 2 small letters. We're going to put them at the top, like so. Now you're going to multiply them into your Punnett square. So remember, we multiply in. So this B times that B, so I get a big B, little b. Do it again, this B times that B, big B, little b. And I do it again for the bottom level, this B and that B, this B and that B. Now this is a nice easy one because as you can see, all my results are the same, right? So let's write out the geno and phenotype. So genotype, 100% big B, small b. See? 1, 2, 3, 4. Or alternatively, you can write 4 out of 4. 4 over 4, like that. Okay? Phenotype. Phenotype, 100%. Now, what color is this cat? If B, big B is dominant, it means our cat's going to be, if we look back here, it's going to be black. Easy, right? Easy peasy. But you know what? I'm going to do a difficult one now. A little bit harder for everybody. Okay? I am going to do a sex-linked disorder because these ones are quite straightforward. Everybody gets these ones right. This is the format, everybody. And to show you, this is how we mark it at the end of the year. We go like this. 1, 2, 3, 4, 5, 6. These questions are often out of 6 marks, and they are easy, easy, easy marks. Okay, I'll do a quick sex-linked one. This will be my last thing for the video today. Okay, guys? So this is sex-linked. Okay, so sex-linked disorder. Let's do red-green color blindness because that's one you have to know off by heart. And yes, if you weren't aware, you have to know it off by heart. In other words, you have to know the lettering. Now, red-green color blindness, we are going to use, let me just put this over here. We are going to use an R, a little r and a big R. Okay, cool. Right, so let's say the question says a male with red-green color blindness has a child with a carrier female. Calculate the chance of having a male with red-green color blindness. Okay? That's the question. I've written it out in full now. This would be something that you may get. So, this is how it goes, everybody. Are you ready? Yet again, we start in the column on the side here. P1. We go back to our question. Now, and you should always be highlighting everything here. Right? Right? So, this is your question. It is a male, and this is an important piece of information. Why? Because this is a sex-linked chromosome. And he has red-green color blindness. He has it. He's having a child with a carrier female. Now I'm going to describe what a carrier is. A carrier female is a female that has one allele for the disorder. But remember, females have two X's, which means that she has a backup allele in case the one is damaged, right? So, here we go. This is how we write it out, and this is how you must write it out. You are going to say male, and this is important. If you don't write the word male for sex-linked disorders, you are going to get it wrong. They're going to completely ignore your question, and I think that's where a lot of you are getting this wrong in the final paper. So, male with, and we're just going to say color blindness. You can even write male with disorder. You can write affected male. They're all alternatives for the same thing. They all mean the same thing, right? Multiplied with, and here's the key, everybody. When you write these crosses, there is no such thing as a carrier female when we do the working out. In the paragraph, we can call her a carrier, but in the working out, we can't call her a carrier. Okay? We can't. So, we call her either an unaffected female, we can call her a normal female, we can call her a normal sighted in terms of normal color sight, but you can't call her a carrier. If it helps you to remember, and this is my little tip, my little hot tip for you when you do these crosses, if it helps you to remember that she's a carrier, use brackets. Because you know what brackets mean to markers at the end of the year? Anything in brackets means I shouldn't mark it, I must ignore it. It's part of their working out, it's part of their thought process. So if you put a word in brackets, the marker, the person marking your work, will overlook it. They'll be like, oh, that's just part of their working out, it's not affecting their answer. And that's a good thing for you. This is going to help you remember that she's a carrier. All right, let's write out their alleles, okay? So males, right? They're X and Y multiplied by our female. She's an X and an X, right? Now let's go in and put in our letters, okay? He's a male with color blindness, which means he'll have a small r on his X, like that. Why do we know that? Because we're going to use big and small letters to show recessive and dominant. On the female, she's a carrier, which means she's got one capital R, one small r, like that. Right, then yet again we go meiosis. We write the word gamete in the margin. Here we go. We separate these. So in one gamete, I have my X with my little r. In my other gamete, I have my Y. In this gamete, I have an X and an R. In this gamete, I have an X and a little r. Okay, we're going to go fertilization. In the column we go F1. We draw our Punnett square, like we normally do. And now we're going to transfer. Remember to keep all the male stuff on one side and all the female stuff on the other side. We don't want them to be overlapped. So we take these two here, X and R, Y, X, big R, X, small r. Now we multiply them in. X, big R, X, small r. I generally put the big letters first. It's more correct. It's not like you're going to get it wrong, but it's more correct to put the big letters first. X, little r, X, little r. Right, next. X, big R, Y, so that's the Y. Y and X, X, little r, Y. Okay, now the question asks, calculate the chance of having a male with red-green color blindness. You now need to answer that, and you need to answer it by writing out the genotype and then the phenotype. So let's quickly do it, okay? So what is the genotype? I have 25% X big R, which is this one over here, X little r. I can't put these two female ones together, hey? Why? Because their alleles are different. Okay? 25% X little r, X little r. Put that in. Let's do the two boys now. 25% X big R, Y. 25%, because that's our one over here, X little r, Y. So I've represented this in some numbers. Let's quickly write the phenotype. I'm just going to have to squeeze it in on the side of the page here quickly. So I'm going to transfer this into the phenotype. So these two females, they have different alleles, right? But their phenotype is the same. In other words, neither of these females are afflicted. So we say 50%, so 25 and 25, right? Unaffected, and you must write female. Oh, squeeze that in on the side there. You must write female. You have to. Okay? Let's look at the men. 25%. Now this is a big R. Does he have the disorder? No, he doesn't. He carries a dominant allele. So 25. And you can say unaffected male. You can say normal male. You can say anything that represents the fact he doesn't have it. And the last one, this male has a small r, which means he does have it. So it's a 25% affected. Oh, I don't think I've spelled affected right. Oh well. Affected male. You can also say red-green colorblind male. And now you've answered the question. So if you go back up here, calculate the chance of having a male with red-green color blindness. What is the chance? The chance is 25%. Okay. I think I'm going to leave it there now, everybody. We've been on for almost an hour now, and I don't want to make this live go on for too long, especially if you're sitting at home. Oh, snap. I just realized. I just saw that now. Somebody corrected me. You are right. There is an affected female. Shooketh. Let me make the change. Sorry, I'm getting so caught up in it. Sorry, everybody. I beg your pardon. So I by accident grouped all of these guys together, all these females together. This is a unaffected female. I beg your pardon. So we've got 25% unaffected female. And then this female has red-green color blindness because she has it on both her chromosomes, okay? So this female doesn't have it because she has a dominant allele that covers or masks the recessive. So that's what our 25% unaffected female is. And then we have a 25% chance of a affected female. Thank you for picking that up. I'm so sorry that I overlooked that. I was going too quickly. Okay. Now, to answer your question, Chelsea, would the unaffected female be a carrier? Yes, she would, but remember, we can't use that word. We're not allowed to use that word when we do the calculations like this, or when we do these kinds of answers at the end. So we call her a carrier when we describe her in the question, and that's the only time you're allowed to call her a carrier. Okay. Otherwise, when you do the working out like this, you have to call her unaffected or normal, or she doesn't have the trait or something like that. Okay. All right. Thank you for making sure I fixed that. Yes, you can put the carrier in the bracket. I think Haley said that. Why? Oh, the question goes by so quickly. Why will a female only be a carrier if it's on both X's? No, she won't be a carrier if it's on both. I just fixed that now. She's a carrier when she's got one capital letter and one small letter. One capital, one small. One dominant, one recessive. Is this Bridget? Did this explain the difference between meiosis and mitosis? I'll do it really quickly. Meiosis makes gametes. Mitosis is for the 3 R's. Repair, replace, reproduce. Meiosis halves the chromosome number. Mitosis maintains the chromosome number. Meiosis makes non-identical cells. Mitosis makes identical cells. Those are the 3 differences. So it's their purpose, which is linked to halving chromosome numbers or keeping them the same. It is when they do their job, making gametes or replacing existing cells. And the third function is the overall linked to making cells that are either identical or non-identical, and effectively making gametes or making just another cell, a muscle cell, a bone cell, or whatever cell. Luanga, oh, you're answering the answers for me. Thank you. Dihybrid. Whoo, guys, I don't have 45 minutes to do a dihybrid. You know how long it takes me to do a dihybrid just in class? It takes me almost an hour and a half lesson to do a dihybrid properly. But I got you. I actually have somewhere here in my notes. I'll give you a quick flash of one. I have a note. Yes, I have lots of handwritten notes. I'm not necessarily making them all available because they are my intellectual property, and some of them are also based off of school notes, and school notes are not my intellectual property, so I can't share things that I didn't create because that's plagiarism. So I'm sure I have a dihybrid here. Give me just but a second and I will show it to you. Where are you hiding, dihybrid? Oh, I see you. Okay, here it is. I'll quickly show you my note and then you can always go back and look at my note in the YouTube. Quick. Okay, here's my note on dihybrid crosses. I had to cross a green or yellow plant, and is it smooth or rough? Let's say it's your leaf. Okay, so I've got a green rough leaf and I've got a green rough leaf. We use big G and little g for our greens, and these are this is heterozygous, rough, heterozygous, heterozygous, heterozygous. I divide them into their respective gametes. This is a whole other video that I would need to explain to you. And then what I do is I put it in this enormous Punnett square and I combine my gametes together. But do you want to know the good news? The good news for dihybrids is they're never going to ask you to draw a whole one, ever. You're going to fill in answers and you're going to answer questions that they have given you. So I don't think you need to be too concerned about them. You're never going to be asked to fill out a full one. Okay. Kiara, do I have a YouTube video? Yes, channel. Yes, I do. The link to my YouTube channel is in my bio on my TikTok. So you can find everything that we've done there. I'm going to post this live on YouTube. Okay, guys, I think I'm going to say goodbye. It's getting quite late in the day. I think we've done a lot in this hour. Some of you have been here from the very beginning. I really appreciate you all. Please go and join my YouTube. I'm going to be posting lots of other things for everybody out there. I'm going to be... Thank you for the TikTok coin. I didn't see who that was. Keeks, thank you. I am going to be posting a lot of stuff, guys. A lot of stuff in the next few days. Yeah, and I'm also, for my own students, going to be posting things that we haven't revised in terms of... I know you're going to see a lot of stuff we've done together already, but I'm also going to put stuff up there that will help everybody. And that was the whole goal of doing all of this, was really to reach out to as many matrics in South Africa. And yeah, so please go and like my videos, join my YouTube, and I will be doing another live very soon and I will let you know. Okay, guys? Thank you. Bye.